Hey guys, we are going to go through the last three sections of gene expression in chapter 16. So we talked about eukaryotes, we're different than prokaryotes, and we talked about epigenetics, and then we talked about transcription, regulation. For those, now we need to talk about post-transcription regulation. Go through these. Post-transcription, here we are. Gene regulation. So we've got transcription, and when we transcribe it, we create the message. We go from DNA to RNA. When we have that RNA, we have post-transcriptional edits that we do. Remember, we talked about we go from the rough draft to the final draft. So that is a way that we can control gene expression. So we have our rough draft here. It's the primary RNA. And then it needs to go into our final draft. And so remember we talked about the spliceosome is going to cut out all of these introns, and we're going to get rid of those via the spliceosome. And then we are going to keep the exons. The exons are going to be expressed, and the exons will go down into our RNA final version over here. We talked about in prokaryotes that translation, transcription, and translation are both happening in the cytoplasm. And so that RNA doesn't have to last very long. It only has to last a couple seconds, like 2 seconds. I think max is like 5 seconds in prokaryotes. Because again, it writes it and then it translates it right after that in the cytoplasm right there in the nucleoid area. For eukaryotes, we have to complete transcription and make that message, and then it needs to leave the nucleus and it needs to go out in the cytoplasm and hit up with the ribosome, so it needs to last longer. We need it to not degrade. So there are other things that we do to the RNA to make it stable. So we are going to add on our cap right here, and we are going to add on our poly A tail. Those are going to enable our RNA to be stable. So now it can go out into the cytoplasm and meet up with the ribosomes and survive longer. The longer an RNA survives out in the cytoplasm, the more proteins get expressed. If you shorten the lifespan of that RNA, then you're not going to make as many proteins. So the more stable it is, the longer it lasts. Whatever protein gets resulted from that happens. When we splice this, something that's kind of cool that eukaryotes can do when we are editing and using the spliceosome and getting out the introns and we're putting the exons together, we might keep and switch around the different exons, that the genes that get expressed. So here we have skipped one. So here's the whole thing over here, and here we're missing the red one over there. So we only kept 2 exons instead of 3 of them. Or we may have some that only go together. They're mutually exclusive. So here we have kept the yellow and purple, and on this one we kept the red and purple next to each other. It just depends on what we got. Here we have alternative, just the blue and purple, and here is all of them. And we have left out this stuff here. There's all sorts of ways that we can take these puzzle pieces and try and fit them together. And when we do, it's called alternative RNA splicing. So lots of different ways. I'm not going to ask you about the different ways, but I do want you to know that there are different ways to assemble RNA, and that it's alternative RNA splicing. One more thing that helps us make sure that that messenger RNA is stable is that we can tack on these binding proteins. They're called RBPs, RNA binding proteins, and those can help with stability. So we've got some here, over here near the tail, and we've got this guy over here. These are, again, RBPs can help increase or decrease stability of that RNA molecule. It just depends. So if we have this RNA molecule and we only want to produce that protein for a short amount of time, maybe we will put on some RBPs that will degrade the RNA after a while. Or maybe we want that RNA molecule to make a ton of protein, and it's got on its cap and its tails, and we're going to tack on some additional RBPs that will help make it last even longer than it would have without the RBPs. The RBPs can decrease the stability or increase the stability. These are RVPs. Other things for RNA stability are microRNAs. And so microRNAs are going to bind to the messenger RNA. So you've made your messenger, you've transcribed your message into the messenger RNA. There are other RNAs out there called microRNAs, and they're short. And what they'll do is they are complementary to the sense RNA, the messenger RNA that you created. They're going to be complementary. So we have this antisense RNA in red, and it can attach on to our sense RNA, our messenger RNA. And if it attaches on to this, now we've got a double-sided RNA molecule, and it's not going to be able to be translated with a ribosome. It won't be able to attach on and run through like it needs to. So in this case, we've created antisense RNA with some microRNAs. Another thing that we can do with microRNAs is besides just blocking it this way and attaching on and making it a double strand, we can do this with the dicer. So we've got, we're going to produce our microRNA, and it is a small one. So this is going to be a perfect match. It's going to meet up with this protein called the RISC. And when it does, it will cause our message to be cut in half. So the dicer will slice that message in half, and now we definitely, we're not going to have translation going on here when we cut our message. Or we're going to have our microRNA attach on, and it meets up with the RISC, and that forms a block to where RNA, the translation cannot come through and run down our messenger RNA like a choo-choo train. It can't do that. The ribosome can't read this like a typewriter. It can't run it through. When the RISC and the microRNA are attached in here, they block it. They're going to repress it. So we can silence it with two ways. We can literally cut it, or we can block it with microRNAs. So something else to look into on gene expression and silencing them. Post-translation regulation. There are different things that we can tack on to under post-translation. If we tack on, say, a phosphate group, we phosphorylate a molecule. Here we have phosphorylated the eukaryotic initiation factor 2 that we need when we undergo translation. If we phosphorylate it, we put on this extra phosphate group that wasn't there before, and it blocks it. So we can't translate in this instance. So that's one more thing that you could do. Other things that you can do, we mentioned methyl and acetyl groups that mess with the epigenetics. They mess with the protein, I'm trying to think of, histones. They mess with the histones, or the methyl groups tack onto the DNA, and that can mess with things. We could tack on a phosphate group like we just saw, and that can block translation, and gene expression can't happen. We can mess, some of these things will shorten the lifespan of a protein. Maybe it's a protein that you really need that is involved in the cell cycle and regulating the cell cycle. Maybe it's a protein that has to do with the cell cycle that we're going to go into when we talk about how we need to stop it and checklist it so cancer cells can't continue on. Maybe it's that protein. And if that protein gets this phosphate on it, it won't work. Or if this protein gets ubiquitin attached onto it, ubiquitin is basically like a red flag that says, I'm done. Even if you're not done, even if that protein is still needed, it pretty much is, it just, you're done. And it throws it into the proteasome, and that's going to dissolve that protein. So it pretty much is an early termination of the protein before it was supposed to go away. So there's all sorts of things that we can tack on and mess with, chemically modify gene expression. So all of these different modifications for accessibility, translation, where they are in the nucleus. Can we get that message out of the nucleus into the cytoplasm where it needs to go? So nuclear shuttling. How stable is that message with RNA? Do we have the cap and the tails? Do we have the binding proteins? All of these things can be modified and mess with translation or post-translation. Cancer and gene expression. We have discussed lots of different steps along the way involving gene expression, from epigenetics from the beginning to post-translation. Every single one of those steps, cancer can get into and mess with. So it's not just one disease, it's not a single disease, it's many different diseases. There's all sorts of ways that cancer can come at this situation and turn this cell into uncontrolled cell growth. So we can mess with the epigenetics. We can make sure that our tumor suppressor gene, maybe that tumor suppressor gene that we need to express, maybe we tap on some methyl groups and now it's locked up with epigenetics and it can't get out, so we can't even express that tumor suppressor gene. Or maybe we put on an acetyl group and we changed our, do we have a picture of it? We changed our proto-oncogenes that tell the cell to grow, and we just kept them, we just cranked them open. We put on acetyl groups and now they are wide open to saying grow, 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 uninhibited, and now they've changed into oncogenes. We could do that. So we can mess with epigenetics, we can mess with transcription, writing those down, messing with the promoter and the enhancer. What if it, again, is something that the cancer needs? It's a protein that the cancer needs. And instead of just having normal growth of the protein, the cancer needs lots of this protein, so it's going to express this gene over and over again. Well, if we are messing with an activator that's going to turn on the transcription and we create more of that activator, that's messing with the transcriptional control. We're turning it on more than it needed to be turned on. And post-transcription, what if we are shorting the lifespan of a protein that we need to regulate cancer, and we're cutting it short when it should have been longer? Post-translation examples they gave of modifying the cell death pathway for colon cancer. They call it the C-flips, and there's two of them. There's a long one and a short one, and both of them are supposed to trigger apoptosis normally. But they can modify the expression of those, and now it says with colon cancer, those colon cancer cells have modified the gene expression to where when they express the long C-flip, instead of controlling the cell growth, it's now increasing the cell growth. Instead of triggering apoptosis, now it's triggering it to grow, grow, grow. So this is an example of post-translation messing with the C-flips in colon cancer cells. Translation, post-translation, targeted therapies. Since we know and we can see, maybe we're doing proteomics, the example in the textbook is about breast cancer. And it says that breast cancer is, what did it say? It's putting out a specific protein. Where did it go? EGF, there it is. The breast cancer cells in breast that are involved with breast cancer are producing this protein EGF in higher quantities. So if you were undergoing proteomics and you're looking at protein and gene expression coming out of someone, and you flagged that this person has this protein, then maybe we could do something about that. And so if cancer is producing this protein, maybe we can stop that, and maybe that will stop the cancer from happening and continuing its growth. So if we could specifically target that protein found in that person's expression of breast cancer, we can do highly targeted therapies. And that's breast cancer. My brother-in-law had testicular cancer. I don't think I've shared this with y'all, but he did. He had it at 21 and stage 4. And he went in remission. We had surgery, everything. We thought he was good. And then it showed back up 2 years ago. And so it showed back up 2 years ago, and they were doing a targeted therapy study trial, trying to look at the expression that is going on in his particular version of prostate cancer, and can they modify it and block that. And so we underwent some chemo to try and change the gene expression happening in his cancer cells, And it turned out that it did. It turned out that the chemo worked. It was the worst chemo ever that people go through. They said it's the worst kind of chemo you can get. And he went through it, but he didn't survive enough to get the treatment. But they did call afterwards and were like, hey, he's a candidate for this treatment, but it was too late. But they do have targeted therapies out there. So there is hope. There's lots of really cool things coming down the line for cancer. So lots of new drugs, lots of ways that we can try and figure out a way to stop this from getting people. Another thing, we talked about phosphates that we could tack things on. So I pulled this picture from earlier in the PowerPoint where you can see if we phosphorylated something, then it's going to pull this guy off, and now the cell growth is triggered. So phosphate groups, acetyl groups, methyl groups, taking our proto-oncogenes and turning them on and turning them into oncogenes all the time. If we mess with our tumor suppressor genes, our P53, that's going to stop the cell cycle. If we mess with it and modified it and it's not doing its job because maybe we put a phosphate group on it, or maybe we blocked its post-translation, something happened and we can't express that gene, then our cell cycle is just going to continue to go on and cancer happens. So lots of different ways. Again, it's not just one incident. It's not just one way that cancer is happening. It's lots of ways. So if we could try and block it at any of these ways, these are all potential therapies to treat cancer. Okay, so we went through all the different sections on gene expression and cancer, and this should finish up chapter 16. If you got questions about 16, let me know. Stop by office hours on Monday, Wednesday.